Rabies. We are still talking about RNA viruses. Rabies virus causes the disease we call rabies. The virus attaches to the nerve cells and goes to the central nervous system. So we're talking about spinal cord and brain degeneration. The important thing to remember about rabies, which is not written here, but you need to know, there are no asymptomatic cases. This is a little bit like, remember smallpox? If you get it, you will get something very bad. And rabies has, I believe, 100% fatality. Smallpox at least had, I don't know what it was, 20% fatality, so 80% survival. Rabies is 100% fatality. So it's very, very dangerous. 100%, it's deadly. And the neurological manifestations, or the symptoms, you could say, typically don't show up right away. Once the symptoms start to show up, then death is near. But there's a really long incubation period, like a month or something like along that line. So, but once the symptoms start, it's a very bad progression. Hydrophobia, I highlighted that symptom, that characteristic. That is common and a very distinctive characteristic, the fear of water. So animals that have rabies and humans have a terrible fear of water, which is interesting. It must be something specific that happens in the brain that affects some kind of part of the brain that is specific for that behavior. Seizures, disorientation, hallucination, paralysis. These all make sense when you understand that it's the brain and the spinal cord that are degenerating. Death results usually from you stop breathing. Respiratory paralysis is the neural activation of ventilation stops. So it's a classic zoonotic disease in mammals, meaning it goes from animal to human, typically not human to human, although if you had a human who was full on with the rabies symptoms and they bit somebody, it could be transmitted, but typically that isn't the way that it's transmitted. So it goes from, in urban areas, city areas, dogs can be a large reservoir, and that's typically where we think of rabies, but actually bats are the reservoir of most cases of rabies that infect humans. So, you could get bit by a dog that has rabies, but you could also be bit by a bat. Or, frankly, any mammal that has rabies could transmit it to a human through a bite. Remember that an animal that is having this brain degeneration may act in unusual ways and may not have fear of humans the way most animals do in the natural world. And so you can have encounters with animals that have rabies that end in a bite. Other ways that you can get rabies, if you had some kind of contact with animal material and the animal had rabies and somehow it got through a cut in your skin, that could happen, or some kind of inhalation, but that's a much less common way of getting it, typically from a bite of an animal that has rabies. They must treat you immediately because, like I said, it has a very long incubation period, and once symptoms start to appear, that's really right towards the end of the disease trajectory. That death is coming soon. So they start treating you even if you get bit by something and you're not sure if the animal had rabies or not. They will give you the treatment because it's 100% deadly. Then they will just give you the treatment, even if they're not sure that you even have contacted rabies. And so if you get bit by an animal, if you can bring the animal to the doctor and they can be sure or test the animal that they eliminate the fact that animal, they can be sure the animal does not have rabies, then you don't need treatment. But if you can't find the animal, or you don't know if the animal had rabies, they will give you the treatment, because otherwise there's a risk of death. And so that's the key thing there. Post-mortem, meaning after individuals have died, if you take sections of their brain, they have these structures that are very characteristic of a rabies infection, and they're called Negri bodies, which are these little dark structures in the brain. That is just confirmation of the fact that that person or that animal died of rabies. So they may or may not be able to find the virions, but if they find the Negri bodies in the brain, that's considered also a confirmation of rabies. Last virus that we're going to look at just briefly is influenza. So we call it the flu, but the full name is influenza. There is a bacterium that has the name influenza in its scientific name, but that was a mistake. They thought that influenza at one point was caused by a bacterial infection, and so they named a bacterium Influenza. It turned out that it does not cause the flu. So that name still is attached to a particular bacterium species, but it's what we call a misnomer. And what that means is it's a mistake. They named it wrong. Influenza, the actual virus that causes the flu, is caused by the virus is called Orthomixovirus. And Orthomixovirus it's a little unusual in that it doesn't just have a single nucleic acid chromosome inside the capsid. It actually has 8 strands of RNA inside the capsid. And so those RNA molecules encode some of the proteins that are on the surface of the virus. And those proteins are called hemagglutinin, HA, and neuraminidase, NA. And both of these are involved in entrance. The hemagglutinin is involved in the ability of the virus to enter the host cells, which are the lung cells. And the neuraminidase is a built-in enzyme that helps the virus to invade in the lungs. So the H for hemagglutinin and the N for neuraminidase, these are proteins that are encoded by the RNA molecules inside the capsid. And so sometimes, well, always, we name different variants of the flu with this H and this N. So a couple years ago, we had a flu called H1N1, and that is a particular variant of the hemagglutinin and a particular variant of the neuraminidase encoding genes. And so because the genes are on these 8 segments, it allows the flu, even more so than any other kind of virus, and all viruses have the ability to mutate and change, but because of the 8 segments, it causes the flu to be just a master at changing and mutating every year. And so the reason that the flu changes every year, it moves around the world in a wave in a very, very predictable way, except for the last couple of years, because I'm making this video fairly shortly after COVID. And because of all the staying home, they did not see flu in the normal patterns the last couple of years, because people stayed home so much that it actually affected transmission of the flu in a positive way. There was much less flu, but typically it comes around the world in a very predictable wave. But as it moves around the world, it changes. And some of the changes are just by plain old vanilla kinds of mutations. So just regular mutation in the RNAs, just changes of the nucleotides, the A's, the U's, the G's, the C's. Those little changes, those little mistakes, those create new, by definition, they create new strains, new mutations, new mutants. A strain is a new mutation of a previous strain. So we could use the term variant, we could use the term strain, we could use the term mutant. They all mean the same thing. And so if the mutations happen in the genes for HA or NA, then we get a new strain. So last year, maybe we had H1N1, maybe this year we'll have H1N2 or whatever. So the numbers signify particular mutation types that are known. That regular plain old mutation mechanism is called antigenic drift. And antigens are the things that are on the surface of the virus. In humans, we have antigens on our cell surfaces. I know viruses aren't cells, but they have things on their surface, so we still call them antigens. So they're kind of like identifying molecules on the surface of something. So the antigens on the flu virus change just by mutation. Now, what's interesting, though, is this other thing. This other thing is called antigenic shift. Antigenic shift is made possible primarily because this virus has multiple segments. For viruses that don't have multiple segments, you could get like a recombinant virus, but not an antigenic shift, not the way that is possible in flu. So let's look at this. On the left side of the screen, we have antigenic drift. That's typical mutations creating a new virus strain. The virus is still fundamentally the same, but a small difference. So here they show a virion shown in green entering a host cell. A mistake during replication changes one little nucleotide, and we might get a slightly different strain when it emerges from that host cell. Antigenic shift, though, that's the thing I really want to draw your attention to. Antigenic shift requires, in order to happen, you would have to have two strains, two different viruses that each have their own versions of H and N. And they co-infect, meaning they both infect the same host and really some of the same cells within the host. And because the chromosome is in 8 segments, when the new viruses emerge, they will probably have some of the segments that came originally from the red virus strain and some of the segments from the blue virus strain. And so you could get this new strain that's really a new combination of these 8 segments compared to what was originally present. So that's considered a new strain when there are combinations of RNA segments from 2 viruses combined together into a new strain. So that's called antigenic shift. That is really made possible by the 8 segments. So with segmented viruses, this antigenic shift is much more likely to happen. With any virus, you can get antigenic drift, new mutations, but only with a segmented virus do you have a high chance of antigenic shift. So make sure you have a very clear definition in your brain, and I typed them up here for you. I think that hopefully helps. And this picture is not terrible, to help you understand what each of those words means.